Welcome back to BioDecode, where biology is decoded into simple steps. This is your trusted audio guide for learning biology the smart way. You're listening to the NCERT Class 11 Biology Audiobook, Unit 2, Structural Organization in Plants and Animals, Chapter 6, Anatomy of Flowering Plants. Before we begin, if you haven't already listened to Chapter 5, Morphology of Flowering Plants and its Exercise Solutions, it's available on our channel. That chapter helped us understand the external structure and form of plants. Now, as we continue in Unit 2, we move deeper, from external morphology to internal structure. In this chapter, we'll study the anatomy of flowering plants, exploring how the tissues inside, roots, stems, leaves, and other organs are organized and how their arrangement supports various physiological functions. You'll learn about tissue systems, including epidermal, ground, and vascular tissues, and discover how monocot and dicot plants differ in their internal structures. This audiobook is designed to make complex anatomical details easy to visualize and understand through clear explanations, logical sequencing, and concise language. Whether you're revising for your school exams or preparing for NEET, you can listen, learn, and build a strong conceptual foundation. So, plug in your earphones, focus your mind, and let's begin exploring the inner world of plant structure. This is BioDecode, and you're listening to Chapter 6, Anatomy of Flowering Plants. You can very easily see the structural similarities and variations in the external morphology of larger living organisms, both plants and animals. Similarly, if we study the internal structure, we find several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. The study of the internal structure of plants is called anatomy. Plants have cells as their basic unit. Cells are organized into tissues, and in turn, tissues are organized into organs. Different organs in a plant show variations in their internal structure. Within angiosperms, the monocots and dicots are also anatomically different. Internal structures also show adaptations to diverse environments. The tissue system. Earlier, we discussed the types of tissues based on the kinds of cells present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function also depend on their location. On the basis of structure and location, there are 3 types of tissue systems. These are, first, the epidermal tissue system. Second, the ground or fundamental tissue system. And third, the vascular or conducting tissue system. Epidermal tissue system. The epidermal tissue system forms the outermost covering of the entire plant body and comprises epidermal cells, stomata, and epidermal appendages such as trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells that form a continuous layer. The epidermis is usually single-layered. Epidermal cells are parenchymatous in nature, containing a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy, thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. They regulate the processes of transpiration and gaseous exchange. Each stoma is composed of 2 bean-shaped cells known as guard cells, which enclose a stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer walls of guard cells, away from the stomatal pore, are thin, while the inner walls, towards the stomatal pore, are thickened. The guard cells possess chloroplasts and regulate the opening and closing of stomata. Sometimes, a few epidermal cells near the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture, guard cells, and the surrounding subsidiary cells together form the stomatal apparatus. Figure 6.1 shows a diagrammatic representation of stomata. In Part A, you can see stomata with bean-shaped guard cells, typical of most dicot leaves. In Part B, it shows stomata with dumbbell-shaped guard cells, as found in grasses. Labels in the diagram include epidermal cells, subsidiary cells, guard cells, chloroplasts, and the stomatal pore in the center. The cells of the epidermis bear a number of hairs. The root hairs are unicellular elongations of the epidermal cells and help in absorbing water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. Trichomes in the shoot system are usually multicellular. They may be branched or unbranched, and either soft or stiff. Some trichomes are even secretory. Trichomes help in preventing water loss due to transpiration. The ground tissue system. All tissues, except the epidermis and vascular bundles, constitute the ground tissue. It consists of simple tissues such as parenchyma, parenchyma, and sclerenchyma. Parenchyma cells are usually present in the cortex, pericycle, pith, and medullary rays of primary stems and roots. In leaves, the ground tissue consists of thin-walled chloroplast-containing cells called mesophyll. The vascular tissue system. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles as shown in Figure 6.2. In dicotyledonous stems, cambium is present between the phloem and xylem. Because of the presence of cambium, such vascular bundles have the ability to form secondary xylem and phloem tissues, and hence are called open vascular bundles. In monocotyledons, the vascular bundles lack cambium. Hence, since they do not form secondary tissues, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged alternately along different radii, the arrangement is called radial, as found in roots. In conjoint types of vascular bundles, the xylem and phloem are located together along the same radius. Such vascular bundles are common in stems and leaves. Usually in conjoint vascular bundles, the phloem lies only on the outer side of the xylem. Figure 6.2 shows various types of vascular bundles. Diagram A represents a radial vascular bundle where xylem and phloem are arranged alternately. Diagram B represents a conjoint closed vascular bundle without cambium between xylem and phloem. Diagram C represents conjoint open vascular bundle with cambium between xylem and phloem. Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization in roots, stems, and leaves, it is convenient to study transverse sections of the mature zones of these organs. Dicotyledonous root. Refer to Figure 6.3a, which shows the transverse section of a sunflower root. The internal tissue organization is as follows. The outermost layer is the epiboma. Many cells of the epiboma protrude outward as unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called the endodermis. It is made up of a single layer of barrel-shaped cells without intercellular spaces. The tangential and radial walls of endodermal cells have a deposition of a water-impermeable, waxy substance known as suberin, forming the Casparian strip. Next to the endodermis lie a few layers of thick-walled parenchymatous cells called pericycle. It is in these pericycle cells that lateral roots and vascular cambium originate during secondary growth. The pith is small or inconspicuous. The parenchymatous cells lying between the xylem and phloem are known as conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, cambium ring develops between the xylem and phloem. All tissues inside the endodermis, that is, pericycle, vascular bundles, and pith together constitute the stelae. Figure 6.3 shows the transverse section of roots. Part A represents a dicot root, showing root hairs, epidermis, cortex, endodermis, pericycle, protoxylum, metaxylum, and phloem arranged radially. Part A represents a monocot root, which shows a similar arrangement but with more xylem bundles and a larger, well-developed pith. Monocotyledonous root. The anatomy of a monocot root is similar to that of a dicot root in many respects, as shown in Figure 6.3b. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. Compared to the dicot root, which has fewer xylem bundles, the monocot root has more than 6 xylem bundles, also called polyarch condition. The pith is large and well developed. Monocotyledonous roots do not undergo secondary growth. Dicotyledonous stem. The transverse section of a typical young dicot stem shows that the epidermis forms the outermost protective layer of the stem, as illustrated in Figure 6.4a. The epidermis is covered with a thin layer of cuticle and may bear trichomes and stomata. The cells arranged in multiple layers between the epidermis and pericycle form the cortex, which consists of 3 subzones. The outer hypodermis is composed of a few layers of collencumatous cells that provide mechanical strength to the young stem. Below the hypodermis, the cortical layers consist of rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The endodermal cells are rich in starch grains, and this layer is also known as the starch sheath. The pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. Figure 6.4 shows the transverse sections of stems. Diagram A represents a dicot stem showing epidermis, hypodermis, parenchyma, endodermis, pericycle, vascular bundles, and pith. Diagram B represents a monocot stem with epidermis, hypodermis, ground tissue, and scattered vascular bundles. Between the vascular bundles, a few layers of radially placed parenchymatous cells form the medullary rays. A large number of vascular bundles are arranged in a ring, which is characteristic of a dicot stem. Each vascular bundle is conjoint, open, and has endarc protoxylum. The central portion of the stem, consisting of rounded parenchymatous cells with large intercellular spaces, forms the pith. Monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis and a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath. It also has a large conspicuous parenchymatous ground tissue, as shown in Figure 6.4b. The vascular bundles are conjoint and closed. Peripheral bundles are generally smaller than the centrally located ones. Phloem parenchyma is absent, and water-containing cavities are present within the vascular bundles. Dorsiventral, a cotyledonous leaf. A vertical section of a dorsiventral leaf through the lamina shows 3 main parts: the epidermis, mesophyll, and vascular system. The epidermis covers both the upper adaxial and lower abaxial surfaces of the leaf and has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial surface, which may even lack stomata. The tissue between the 2 epidermal layers is the mesophyll, made of parenchyma cells containing chloroplasts that carry out photosynthesis. It has 2 types of cells, the palisade parenchyma and spongy parenchyma. The adaxially placed palisade cells are elongated and arranged vertically and parallel to each other, while the spongy parenchyma lies below them with large air spaces. Figure 6.5 shows the transverse section of leaves. Part A represents a dicot leaf showing adaxial and abaxial epidermis, mesophyll with palisade and spongy cells, and vascular bundles. Part B represents a monocot leaf, showing uniform mesophyll tissue without separation into palisade and spongy layers. The vascular system includes vascular bundles, seen in the veins and midrib. The size of vascular bundles depends on the size of veins, which vary in thickness in the reticulate venation of dicot leaves. Vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Isobilateral monocotyledonous leaf. The anatomy of an isobilateral leaf is similar to that of a dorsiventral leaf, but shows distinct differences. In an isobilateral leaf, stomata are present on both surfaces of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma, as shown in Figure 6.5b. In grasses, some adaxial epidermal cells along the veins become large, empty, and colorless. These are called bulliform cells. When bulliform cells absorb water and become turgid, the leaf surface spreads out. When they lose water due to drought or stress, they become flaccid, causing the leaves to curl inward to reduce water loss. The parallel venation in monocot leaves is evident in the nearly similar size of vascular bundles, except in the main veins. Summary. Anatomically, plants are made of different kinds of tissues. Plant tissues are broadly classified into meristematic, including apical, lateral, and intercalary, and permanent, which may be simple or complex. Their major functions include the assimilation and storage of food, transport of water, minerals, and photosynthase, and mechanical support. There are three main tissue systems: epidermal, ground, and vascular. The epidermal system includes epidermal cells, stomata, and epidermal appendages. The ground tissue system forms the bulk of the plant and is divided into three zones: cortex, pericycle, and pith. The vascular tissue system is composed of xylem and phloem. Depending on the presence or absence of cambium, vascular bundles may be open or closed. These bundles conduct water, minerals, and food material. Monocotyledonous and dicotyledonous plants differ in their internal structures, particularly in the number, type, and arrangement of vascular bundles. Secondary growth occurs in most dicot roots and stems. You've just finished listening to Chapter 6, Anatomy of Flowering Plants from NCERT Class 11 Biology, here on BioDecode. I hope this audio helped you clearly understand how the internal organization of tissues, from the epidermal layer to vascular bundles, defines the structure and function of roots, stems, and leaves. We also explored the differences between monocotyledonous and dicotyledonous plants, the three main tissue systems, and the remarkable adaptations that allow plants to thrive in diverse environments. In the next episode, we'll go through the exercise solutions and important questions from this chapter, explaining each answer step by step to help you prepare confidently for your exams. So make sure to follow BioDecode and stay tuned for the next part of this series. Keep learning, exploring, and I'll meet you in the next audiobook, Chapter 6, Anatomy of Flowering Plants, NCERT Solutions and Key Questions. And yes, students, don't forget to like, share, and subscribe to our channel, BioDecode. See you in the next audio lecture.